This PowerPoint is going to cover the second part of Unit 1 in terms of the AP Biology test, remembering that all of Unit 1, which also includes 1.1 and 1.2 related to chemistry of life, is going to cover 8 to 11% of the AP Biology test. This PowerPoint is related all to biomolecules. So things that you're going to need to know. We're going to learn about the process called hydrolysis and dehydration synthesis, and I will break those down for you in a little bit, but those are two processes that we use to stick biological molecules together and break them down. Things that you're not going to need to know. So they've kind of relaxed a little bit on some of the extreme details for the test. You're not going to need to have to draw out any amino acids. You're not going to have to know the structure of specific nucleotides or carbohydrates. It's more about understanding the general processes and using that information. Kind of like a, SAT, ACT passage. Other things you need to know. That directionality is important and that the direction that things are structured is important for its function. So for example, nucleic acids like DNA and RNA have a specific 5' to 3' direction with specific base pairing rules, and we'll get into that. Proteins and amino acids, depending on how they're organized and which ones are in what order, that affects how they fold. And therefore, depending on how they fold, it'll affect their function. And it's totally the same thing with carbohydrates, of whether they are a line or a branch, things like that. This is kind of the general idea of structure leads to function. And finally, for 1.6, you're going to need to know that DNA and RNA have similarities and differences. So just being able to kind of compare and contrast the two. Okay, so what is a biomolecule? It's basically a subcomponent of life, and the sequence of it determines its properties. So we're going to go through the major components of biomolecules. To understand this, we need some vocab. So mono as a prefix means one. So a monomer is a single subunit, whether it's a single carbohydrate, a single sugar of that carbohydrate, a single amino acid, any of those things. And then when you begin to put them together, you create a polymer. Poly means many. So really, if you start sticking them together, it creates a polymer. And that process is called polymerization. And depending on how that is lined up, they fold and create different structures. So on the left, we have a starch with a more branchy structure. On the right, we have a starch and a different structure. And because of that, they have different properties. Monomers are assembled into polymers by what's called a condensation reaction. And really the important vocab here is going to be dehydration synthesis. And if you look at that word, to synthesize something means to make. So I always think of photosynthesis because that means to make food with light, which plants do. To synthesize something means to make. So we are making a polymer from a monomer. Dehydration, if you're dehydrated, that means you don't have enough water. So the overall process means that you are making something by taking water away. And if you think about and look at the picture down here, that's essentially what's happening. The end of one chain here has a hydrogen, and the end of another has an OH. And because of that, those two things basically stick together and make water, allowing the molecules then to stick together. So this monomer kind of gets added to the chain. And then another one can come along and another one can come along. And that's how we build a large chain. So dehydration synthesis, again, just means to make something by taking water away. On the flip side, if taking water away joins things, well, then adding water is going to split them up. So hydrolysis or hydrolysis, to lyse something means to break it. So we are breaking a bond with water is what is happening. So it's just the opposite of dehydration synthesis. Nucleic acids. Those are DNA and RNA. Those are the things that make us. And the building block of nucleic acids are what we call nucleotides. So a nucleotide, here's a big long chain of DNA, for example, and here's one side of that twisted ladder. If we zoom in, a nucleotide contains 3 signature pieces. There's a sugar group. In this case, if it's DNA, it's deoxyribose. If it's RNA, it's ribose. A phosphate group. And these 2 things alternate and form the sides of the ladder. And then the third component is a nitrogenous base, which form the rungs of the ladder. There are 4 nitrogen bases that we will learn about: adenine, thymine, cytosine, and guanine, ATCG, for DNA anyway. So those 3 components together make a nucleotide, and when you start linking them all together, they can form a nucleic acid. So here's just a zoomed in photo of what I had just mentioned in those basic components. In this case, the nitrogen base is adenine, A. In terms of comparing and contrasting, the sugar is slightly different from DNA to RNA. So in RNA, it's ribose sugar. And you can see that structurally these are identical except for this spot right here, where this one has an oxygen. And when you take that oxygen away, we get deoxyribose, which makes sense, right? Because it's ribose minus an oxygen, so deoxyribose. So if you're using this one on the left, you are creating DNA, and if you're using the one on the right, you're creating RNA. Otherwise, structurally, they are the same. I'm not sure if you're really going to need to know this specifically, but they do form bonds. The alternating sugar and phosphate groups on the side of the ladder here do come together, and they form what's called a phosphodiester linkage. It just is a way in which to join the sugar group with the phosphate group, and then that happens repeatedly as you move down. And similarly, this happens just as we had mentioned earlier. When monomers turn into polymers, we need to take away a water. This is a dehydration synthesis reaction. That bond forms when you remove a water. Summing it up, DNA has deoxyribose, as I mentioned. RNA has ribose. Other things that I didn't touch on: DNA has the nitrogenous base thymine, or T. In RNA, it's a little bit different. Instead of T, they use U, or uracil. And we'll get into the details later when we talk about how we make proteins and things, but that's one structural difference. And then lastly, the shape. DNA is in a double helix or twisted ladder form, whereas RNA is a single helix. It's just one side of the ladder with rungs. We are now going to move on to proteins. Proteins are derived from polypeptides, which is basically a chain of amino acids. And you can see here a chart of the 20 amino acids. There's since been more, but you can get the idea of what those different amino acids are. And depending on the structure, they have different properties. In proteins, the order determines the shape. So as I showed you on the previous slide, some of these are nonpolar, some of them are polar, meaning they have a slight charge. Some of them are acidic, some of them are basic, meaning, again, they have a different kind of charge. Some of them like water, some of them don't. So depending on the chain and the properties of each one of those amino acids in the chain, they're going to want to be close to each other or not close to each other, hide on the inside away from water, or love the water and be on the outside. So that's going to determine the overall shape. And then when they do take on a shape, they fold together and they can join. For example, this would be the 3D structure that it creates. it then has a certain function. So this is an example of a functional protein, hemoglobin. It actually has a couple of different chains that ended up sticking together, but this is what it would look like in 3D form. Every amino acid has a basic structure. They all have an amino group, which the amino is really for the nitrogen. So there's always this NH2 on one side, and there's always a carboxyl group on this side. So carboxyl is COOH when you get to organic chemistry someday. And then there's always going to be a hydrogen here. So these three components are always going to be a part of an amino acid. There's an amino group, a carboxyl group, and an H. The R group is what's different. So in the giant table of amino acids, the R group changes and gives it the different properties. The rest of it is all the same. And that's the basic building block of a protein. The R groups in this case are also called side chains. They're highlighted in white here so you can get an example of some of the different structures that they can have. But you can see that the lower part that's not in white is all the same for each one of them. Amino acids, when they are linked together, it creates a peptide bond, which is why when you put a bunch of amino acids together, it's called a polypeptide. Just like before, we're putting them together. We are synthesizing a polymer, so we need to do dehydration synthesis. So we kick off a water. So the OH from the carboxyl group and an H from the amino group come together and make water, and then that allows the two to stick together. And then you get this super long chain. That's what we call the primary structure. So it's just one long line of amino acids. And then they create what's called a secondary structure. So the hydrogen bonds, which remember are from the slightly positive and negatively charged parts of certain molecules, they create either an alpha helix, which is kind of a circular or a ribbon shape, or a pleated sheet, which looks, as the name describes, a pleat. So this is an example of secondary structure as a result of hydrogen bonds. And then that secondary structure folds together and creates what's called tertiary structure. So this could be hydrogen bonds. It also includes, as I mentioned, hydrophobic ones don't like water, so they're going to want to hide themselves on the inside. Hydrophilic ones are going to want to be on the outside. Sulfurs love to form what's called a disulfide bridge, so they love to bond super strongly together. So if there's two in the near vicinity, they like to hook up together, which is why we ended up kind of swinging this tail this way instead of having it be a line. But this is tertiary structure. And then finally, like the hemoglobin molecule earlier, we get it all put together in this quaternary structure, and that forms the final molecule. And this structure is really what gives it its function. Another example, as shown on the left here, is collagen. So this is three different proteins that are joined together kind of like a twisted rope, and then the hemoglobin one I had mentioned. Other examples of proteins that are going to be super important throughout this course, enzymes are proteins, so they are responsible for speeding up chemical reactions without getting used up. We have defensive proteins, so for example, antibodies are proteins. Storage proteins, so for example, the milk protein is one for storing. And finally, transport proteins are super important. So like hemoglobin is one example that likes to carry iron throughout your body. Proteins are one of the number one things that our body uses in a diverse set of ways to kind of keep us alive. We have hormone proteins. So insulin, if you've learned about diabetes, is one example. Receptor proteins, so allowing you to respond to certain signals from other cells. There are movement proteins, so allowing us to contract and relax our muscles. And then collagen, which was mentioned before, is an example of a structural protein. We also have keratin in our hair and nails and things like that. If the protein loses its structure and therefore loses its function, it's been said to be denatured. If something is denatured, it can't work anymore. Essentially, you've broken it. So proteins like to live in a happy temperature / pH salt environment. So different ones inside our body are meant to work best at, body temperature, or the ones in our stomach are meant to be at an acidic pH of our stomach versus the slightly basic pH of our intestines. They have a happy kind of optimal level. And when you start to go outside of that, then that's when you start messing with the proteins so they don't work anymore. And if they're not structured appropriately, they're not functioning appropriately. Next, we have lipids. Lipids are ones that do not form polymers. So they're kind of the exception to that rule. But I always make lipids synonymous with fats. Phospholipids, which is what the cell membrane is made out of, and also steroids. So here's an example of pictures at the bottom of what all of those look like. Triglyceride, meaning that it has 3 here. And steroid has to do with the ring-like structure, and a phospholipid because it has a phosphate molecule here stuck to the side. In general, they don't have a charge. They don't really like interacting with water, which is why oil and water or fats and water don't like to mix. Lipids themselves are hydrophobic. So again, they form non-polar covalent bonds with one another, and they like to hide away from water. So here's an example of a hydrocarbon chain. They can be saturated or unsaturated. What that really means is it saturated with hydrogens? Meaning, does every available carbon in the chain have hydrogens all around it? If so, then it's called saturated, and it makes a single straight line. If it's unsaturated, that means that there are some hydrogens missing. You could always add more. And what that means is when you take the hydrogens away, you make the bond between the carbons a little stronger. And therefore, it causes a kink. So unsaturated fatty acids have this little kink. Saturated fatty acids don't. And because of that structural difference, then that leads to different functional properties. Another thing about this is fats are constructed from 2 things. We have the fatty acids, which are down here at the bottom. And there's also usually a glycerol molecule. And this is what glycerol looks like, and it kind of shows you how it would come together. So the glycerol comes here with the fatty acids to make this triglyceride. And you see, again, we're synthesizing, so water is given off in the process. All right, fatty acids are linked together in what we call an ester bond. You don't need to know the specifics, I don't believe, but I just wanted to show you what that looks like. As I mentioned, we're synthesizing, so this is again a dehydration synthesis reaction. We're sticking things together with the loss of water. And here in highlighting, you can see where the 2 H's and the O are coming from. And depending on if it's saturated or unsaturated, we'll give it its functional properties. The saturated ones are the straight lines, and therefore they can get a little bit closer together. So because of that, they like to form solids at room temperature. Butter is an excellent example of a saturated fatty acid. Whereas if they have those little kinks, because they don't have a total number of hydrogens around them, that means they can't quite pack in as closely with one another, and that often results in a liquid at room temperature. So oil is an excellent example. Phospholipids, again, what the plasma membrane is made out of, have polar regions at the top. So this is a charged little area here at the top that likes to interact with water. And then they also have a non-polar region, which are the tails, that don't like to interact with water. And as we get to the plasma membrane, you'll see that these heads like to be on the outside by the water, and these tails like to hide on the inside, away from water. Finally, we're going to talk about carbohydrates. These are your sugars. And so here's an example of some chains of each one of those sugars linked together. A singular sugar by itself is called a monosaccharide. Mono meaning one, and then saccharide is just the term that means sugar. When we start to string them together, we get a polysaccharide. Monosaccharides have a basic formula. So it's some multiple of this, CH2O. So the most common one I would say is glucose, and that would be C6H12O6. And here's what its structure looks like. You can see that almost all of them have this ring-like structure of 6 carbons in this case. When we start to put them together, then we create what's called a glycosidic linkage. So again, we're linking together, so it's dehydration synthesis. We take the OH from one and an H from another, and we create water. And we start to form, this would be a disaccharide because it has 2 of them. So we have monosaccharides and disaccharides, and then everything more than that would be a polysaccharide. In general, the functional role of these things is basically structure. So they maintain structure, and they also store energy. So like glucose and fructose, this is kind of our energy stores. It's sugar. It's why we eat it, so that we can get energy. In this case, you're seeing glucose and fructose coming together to make the disaccharide sucrose, noting that they all end in ose, which is sugar. Starch is kind of the main storage polysaccharide in plants. So it has a long chain of alpha glucose molecules. There's also a beta glucose molecule, but we don't really need to get into that per se. It's just in how the molecules happen to be arranged. But when you link them all together, we create starch. Glycogen is the storage polysaccharide in animals. So this comes from crazy high branched chains of alpha glucose. So this is mainly stored in your liver, in your muscles, but this is kind of that energy storage. So when we need energy, we eat glucose, we eat sugar, and then our body can then break that down using cellular respiration. We break down glucose. But we don't always need all the glucose that we eat at the time. So a lot of times we store it as glycogen. And in order to do that, we take all those glucose molecules and kind of branch it up, and we package it away in this form. And then if we need it later, we can start to break this down into usable parts to make energy. Cellulose is the component of plant cell walls. So instead of alpha glucose, this is a long chain of beta glucose, and that's the structural component. Whereas before we saw starch as kind of the main food storage component for plants, cellulose is the main structural component for plants. As you can see on the top, that's the long chain of alpha glucose molecules, and on the bottom it's the beta glucose molecules. And really they are the same thing, it just has to do with where the molecules are oriented. So you can see in the alpha, it's just the exact same thing all the way down. In beta, it kind of alternates. Another one to talk about is chitin, and this is another structural thing that's found in the exoskeleton of arthropods, like grasshoppers. It's also in the cell walls of fungi. But you can see it's just another example of a structural component and what that looks like. Similar to all the biomolecules, directionality is important. So, for example, DNA is always going to be going in the 5' to 3' direction. We'll learn more about it later. And then the opposing strand, or what we will call the complementary strand, always goes in the opposite, 5' to 3'. So this left side kind of goes from top to bottom. The right side goes from bottom to top. Proteins have directionality as well. The head, so to speak, is the amino group, or the top part, and then the tail of it is always going to be the carboxyl end. So we'll learn that proteins always start with the very same amino acid, methionine, and then always have variable endings with one of the terminal amino acids, but there's always a head and a tail, so directionality is important. The types of bonds determine their orientation. So depending on how they're stuck together, that can then show what they turn into in a 3D type structure. And just tying it back to general big picture, structure leads to function. So if you start to change the structure, it can't do its job as effectively, so you're going to change the function. Sickle cell is one very clear example of that, where there's one change. So the primary structure is a series of amino acids in this protein. And then number 6, for example, gets mutated or changed in some way. So instead of GLU, it turns into a valine, VAL. And then that, when it folds up, because it has a different property, starts to look like this. And therefore, when it goes to make its larger structure, it can't quite form correctly, and therefore it doesn't hold oxygen as well, and you end up with a final structure of this. So that one small change in structure leads to a large change in function. Okay, so final review. Amino acids, it shows you the basic structure, examples of where we would find them, and functions. Same thing with a nitrogenous base. There's a nucleotide, for example. There are the 3 basic components. We have DNA and RNA as the 2 main examples in what they do. Carbohydrates, this is an example of the basic building block of monosaccharide. We can see that there are monosaccharides, disaccharides, and polysaccharides and what their jobs are. And then we also have the lipids down here. So different types of lipids, examples, and then their jobs. So I think overall it's important to just know the major classes, where we would find them, what process we use to link them together, to break them apart, and what their functions are. And that's it.